Okay. Yes, I think we're going to get started. It's a pleasure to have all of you guys here. It's with a great pleasure. I'd like to introduce Dr. Jürgen Cox as the today's webinar presenter. Dr. Cox is going to talk about multiplexing with isotopic labels for DDA and DIA in MaxQuant. Um, a couple of kind of logistics. If you have any questions, you can kind of raise your hand. I will open up your microphone, and you'll be able to directly chat with speak with Dr. Cox during the webinar. Or if you can hold until the end of the webinar, you can just wait or put your questions in the chat. Then, at the end of the webinar, we'll answer those questions. A quick introduction about Dr. Cox. He has done his diploma in physics from RWTH Aachen University in Germany. And then he spent his time in the US at MIT, getting his PhD in physics. Worked in industry for some time at Gene Data and as a postdoctoral researcher at Technical University of Munich. And after that, again, went back to Gene Data and came back to academia again. So he has a good mix of academia and industry exposure. Currently, he's a research group leader at Max Planck Institute of Biochemistry. He also holds many honors and awards. Primarily, the main ones are the German Mass Spectrometry Award in Life Sciences and Gilbert S. Omen Computational Proteomics Award from USOPO. Thank you so much for Dr. Cox. Please take it away.
Yeah, so hello everybody, and thank you very much to the Silantes team for inviting me. And thanks to all of you for showing interest and joining the webinar. So indeed, today it's no surprise that I will be talking about MaxQuant probably. And in particular, I will be talking about the uses of isotopic labeled data in MaxQuant. And so since long, we can deal with that in the form of DDA data, for example, SILAC, DDA labeled data. But also recently, we also support DIA data together with isotopic or any kind of label, could also be chemical labels. And so this is actually what I will be focusing on today, because probably most of you know how to use or that MaxQuant can be used for DDA SILAC data. And so we started about 3 years ago actually extending MaxQuant, that is the well-known DDA platform, also to a data-independent acquisition. And so the idea here was that so for many of the things that are established in the DDA world, in the MaxQuant algorithm portfolio, that we can also reuse them in DIA. And so this is like a peptide-centric DIA workflow that we support here. And in this workflow, you need many computational parts that you just in the same way use in DDA. As, for example, everything that happens on the protein level, like, for example, protein grouping or applying of FDR or pulse discovery rate on the protein level. This is by no means different if the data is DDA or DIA. Right? So one can just take the trusted algorithms over and use them here as well. And then, of course, there are many things that are unique to the DIA workflow that we had to program from scratch or modify significantly from the DDA counterparts. So, okay, so what exactly are the topics of today? So there are 5 things that I want to talk about that are recent additions to the MaxQuant DIA workflow, and that in particular also pertain to the multiplexed DIA data analysis in MaxQuant. And so these are these 5 things here. So that's the on-the-fly library prediction. So it's about being able to do the DIA data analysis completely without any libraries, not even predicted libraries. You have to specify, because they're all going to be predicted from the peptides that are in silico digested from the FASTA file, and then predicted by machine learning. Then indeed, so I go a bit into details of which kind of multiplex labels you can use in MaxQuant, and I can give it away already now. So it's essentially all of them basically, and also how we exactly deal with the multiplex labels within the workflow. Then I'm going to introduce the MaxLFQ workflow for relative label-free quantification of multiplexed DIA data, which is an extension of the algorithm that is also in long use already in the DDA MaxQuant. And then there are two remaining topics that are there to improve the data for quantification. The first one is sequence coverage increase. So this is basically another search after the main search that is there for identifying more peptides on the already identified proteins with a fixed protein FGR, for example, 1%. And then finally, I talk about peptide correlation filtering. So this is basically looking at the peptides that belong to a protein and looking at their protein abundance profiles over the different samples, and if they correlate, and basically to get rid of those peptides that show a significantly different intensity profile over the samples than the majority or the cluster center of all the peptides that belong to a protein. So let's get into the first of these points. So the on-the-fly library prediction. So what happened so far in MaxQuant is that you had to download in silico predicted libraries from a server, basically together with the FASTA files. And these were produced by a recurrent neural network. So these were like complete proteome predictions of the whole UniProt files, basically. However, you had to download them, and you were a little bit restricted in that sense, because you could not just come with your own FASTA file with specific, let's say, for example, derived from a personalized genome or something like this, or any application you might think of. But you had to it was difficult to do this. In most cases, you had to use these standard proteomes for doing your DIA search, right? And so now this is different. Now you just specify any FASTA file, and this in silico library is then predicted on the fly, and we use not the recurrent neural network anymore, but the so-called XGBoost algorithm that's basically trained on sequence window features. And as I said, it cannot be any in silico digestion of any FASTA file you like to use for this purpose. So maybe just to recap what kind of prediction algorithms for the library you can have in general. So they can be roughly subdivided into 3 parts. So the first part you see on the left is like the deep learning algorithms, for example, recurrent neural network algorithm that basically are in their nature able to deal with peptides of any length. So this is for conventional machine learning a problem. If you have like variable length input, you always want to have vectors with a fixed length. But so recurrent neural networks in particular are actually built for being able to deal with these variable length peptides, just as they're able to deal with sentences with a variable number of words in them, basically, right? So that's why they're very suitable and make very good predictions. Then another way to get around of this just take the pointer of this variable sequence length is to just restrict yourself always to peptides of a certain length, like for example, length 7 here, length 8, length 9, and so on, and build separate models, for example, support vector machine models for each of them separately, and then basically you have an ensemble of models, one for each length of peptide sequence, which of course has the problem that there is no synergy between the different models. And then the final one, so that we are pursuing here, is that we take a sequence window approach, basically that we make predictions of the spectral intensities for each bond separately. And then as a feature vector, we take like windows from from minus whatever n to plus n of this bond, and one encode the features within these windows and make a prediction based on this. And there's some metadata, like the total charge of the peptides and the amino acids on the termini and so on, added to these windows. And so this is the approach that we are following with XMOS. So we ship a model together with MaxQuant, so that then you really don't need to do anything, but you can also train your own model. So let's say you have some peculiarities, be it from the mass spectrometer or be it from the wet lab part or from the experimental design, that makes your spectra look different from the spectra that were used to train the default model. Then you can train your own model. So this is like a pop-up window within the MaxQuant software, where you can then specify the so-called MSMS text files that you obtained in a DDA search, but they can also be exported from another DIA search, actually. And they are then used to train this XMOS model, and you can also specify some limited number of modifications, and you will have your customized prediction model. For example, if you're working on your own chemical label or something like this, that might actually change the fragmentation patterns of your spectral library, then you can actually take this into account by building your own custom model. So again, what are commonalities and what are differences between DeepMass and XMOS? So both of them predict not only Y and B, so we're talking about HCD and CID here, right? So Y and B you take for granted, but so we also predict the neutral losses of each of the two series. So that means water losses or ammonia losses, and A2 is also predicted for each of them. And then we have a separate trained model, one for HCD and one for CID, right? And so let me first say that if one wants now to measure the quality of the prediction, there are generally two ways of doing this, which are quite different. So one is to just compare the predicted spectra with the true spectra in cases where you have like the ground truth. So this could, for example, be that you put away test data that you don't use for training, or you could do cross-validation, so different ways of doing this kind. And then you can just measure the spectral angle between the predicted and the true spectrum. Another completely different way of measuring the quality of the library is to just monitor how many peptides and proteins do you identify when using this or that predicted spectral library, for example, differing by using different parameters during the training. And so both of them are valid benchmarks to test the quality of the library. And so we do both of them. And what we found to our surprise is that they don't 100% coincide. So it could actually be that the optimal parameter set for the first criterion is actually slightly different from the optimal set of parameters for the second criterion. And it's actually something that we see in our data. And so what are actually the parameters of the XMOS algorithm that can be optimized? So first of all, these are all the parameters that are relating to the XGBoost algorithm itself. So XGBoost is a tree-based method that is very popular for doing machine learning nowadays. And then one can optimize the window sizes for the Y and B series ions. And then we have an independent window size that is independently optimized for the two neutral loss series. So then, yeah, we optimize both of them. And what you can see is actually that you can actually obtain quality of prediction with XGBoost that actually gets close to the quality of prediction of recurrent neural network-based prediction. And so far so good. And what we also do is we have new prediction models for the retention time prediction, and if it applies for the timsTOF Pro, also for the mobility. And there we also use now XGBoost-based models as opposed to the recurrent neural networks that we used before. And this actually improved the identification rates when we switched from the recurrent neural networks for the retention time prediction to the XGBoost-based model. Right. So then, natural question is, which kind of labels can you use for the data analysis? So you can actually, so simple answer is essentially any kind of label you could think of. So this includes also all kinds of isotopic labels, and you can see here in the interface of MaxQuant, just as as you may know it from DDA data, you can just say what is the number of labels, and then for each of these labels, like if you select 3 here, then for the light, for the medium, and for the heavy, you can then specify separately what exactly the molecular composition of these labels is, and they can be either isotopic, but they can also be chemical, so then they can be of any kind. And so, for example, if you would be working yourself on some new kind of labels, there would be no problem in including them here. You would just have to define the composition of these labels and select them here accordingly. And so what then happens is for the quantification later, so MaxQuant actually uses both MS1 and MS2 for the quantification. And so for certain kinds of labelings, you cannot use all MS2 features because they are identical for the different labeling states, right? So depending on how you label N termini and C termini, you might have identical fragments for one of the sides. And then, of course, you cannot use them for relative quantification between the different labeling states. But MaxQuant detects this automatically from the definition of the labels, which sides, or maybe both sides, it can use for doing the quantification. So then the next step is to identify the spectra in the multiplexed DIA sample, and for this we use a multiplexed in silico library. So we search by default for all of the labeling states, but you could also restrict the search for only one of the labeling states or any subset of the labeling states, which might sometimes be better if you have a reference state. Like, let's say your heavy channel is always a carrier channel that may be more abundant, and the others are, let's say, single cell channels. Then you could also say, okay, I only do the library search for the carrier channel and I'm not looking with the library search for the other channel. So that is absolutely possible to do, but it's so in general, basically the data type of the library and the data type of the DIA sample is completely decoupled. So it could be completely different labeling, anything. So actually MaxQuant does not take any information from the library like pre-calculated masses or so. So everything is calculated from scratch based on the specified information. So then the next step, so after we have identified now the features by matching to the spectral library, the next step of identification is to find complete MS1 multiplexes. So what does that mean? So for example, if you use triple labeling, so you have, like, for example, SILAC, light, medium, and heavy, MaxQuant will now only look at the MS1 data, and it will try to find these complete triplets of light, medium, and heavy peaks. And basically, so discarding the MS2 data, it will then know that these MS1 features belong together in a triplet. And if one of them is identified, let's say the left one is identified by MSMS, then it knows that the medium and the heavy ones here can be used for the relative quantification between the 3 channels without having identified the other ones with MSMS, right? So that is the idea, that you have, like, just based on MS1 feature pairing, you increase the basis for the quantification, so completely independent of the MS2 here. And but still, the restriction is here that these features have been identified as isotope patterns, as 3D isotope patterns in the MS1 data. So you might still miss some of the signal in certain channels. And so this you will get now with the so-called requantify algorithm, which is actually exactly the same as we also used in DDA data. And so the idea of the requantify algorithm is that if you identify a certain isotope pattern by MS and MS2 spectra, then it looks for, and let's say this is not paired up with other features on the MS1 level, then MaxQuant or the requantify algorithm will actually look for the place in the data where you would expect the heavy partner to be, and it would then integrate over the data, over the available raw data there, and by that you would get a quantification of the data as well. So that might then be underestimating the ratio because so it's a very faint signal that actually by itself did not lead to an MS1 isotope pattern detection, but you might with that pick up a good estimate for very extreme ratios as well. So exactly the same as you know it from DDA MaxQuant. So then finally, not finally, so next thing you need is actually then this label-free quantification algorithm, and there we extended the MaxLFQ algorithm that you also may know from DDA already, and which consists of two parts. One is the normalization and one is the protein quantification part. Normalization being relatively straightforward of bringing like the intensity distributions to comparable scales, and the protein quantification kind of without going too much into details, following the idea of first making all possible pairwise comparisons between samples, so between sampled pairs, and then in each pairwise comparison, taking the maximum amount of peptides that you can use for doing this pairwise ratio. So basically taking all the peptides that are in common between these two samples, and then by some intricate median calculation, reporting a pairwise ratio, and then going from this triangular matrix of pairwise ratios by some optimization to an intensity profile that kind of tries to extract the most optimal quantification profile from this pairwise distance matrix. And so this, if you now go from DDA to DIA, the advantage is that you now have more independent data than you have in DDA. So in DDA, you were only using the MS1 features, but here now we can also use the MS2 features. So, so by just going to DIA, we have like a 10-fold increase on average of the features that you can use or that you can put into this MaxLFQ algorithm, and they're basically all put as independent channels. And in each pairwise comparisons, you look if either the two precursors or the precursors are both there in both samples, and for each fragment type separately, like for example, B3, you look if this B3 is present in both of the samples, and then you can take the log ratio in that case for the B3, and you can do this for the precursors and for all of the fragments, and then by that taking a median ratio, log ratio over many more single ratios. So then this is for unlabeled DIA. So how do we deal with this then if we have labels? So very straightforwardly, so if we have labeling in DIA, so again, any kind of label, we treat each label in each LCMS join as a separate sample. So what does that mean? So first of all, they're all equally, and we'll get a very simple to use matrix out of like protein quantification matrix out of this, where you just where you don't have to distinguish between within sample and between sample comparisons, but you just have, or let me show this example. If you have like 10 LCMS runs and you have 3 labels, that would mean you would just get a matrix that looks like you have 30 samples. I mean, that's also indeed what you have. And you get like 30 columns with LFQ intensity. So this is then very easy to proceed to the next step and do your t-test and clustering on this kind of data. So the results are again LFQ intensities, so not ratios between heavy light or something like this, although we also provide these, but this is not the main result, and you find them in the protein groups.txt file as you would find it for DDA results. So just while I'm coming to an end, just to show you some results. So this is data that was provided by the lab of Matthias Selbach in Berlin, where he used like double SILAC labeling, so with the heavy and light channel, and using the heavy channel as a reference, and then the light channel has a species mixture between E. coli and human in different ratios, so that you can produce many benchmark ratios with known ground truth. And so what we get out of the MaxLFQ calculation you can see here for different kind of expected ratios that you see on the right side here. The box plots are indicating the range of ratios that we get for the whole proteome there for the whole E. coli proteome, and they are then put against the expected ratio, and you see like a very good coincidence between the expected and the true ratios here in this case, and also good relatively narrow quantification for each of them. Then so the final or the next feature is sequence coverage increase. So what does this mean? So if you run MaxDIA, it will run through the whole workflow up to the protein group FDR. And then after the protein groups are extracted with 1% FDR and also using PSM FDR of 1%, we do another search, and now we use a restricted FASTA file that only contains the proteins that were identified with this 1% protein FDR, and we perform a new search with a new peptide level FDR calculation, which you can choose yourself. But so we have, in the example, we have fixed it now to 5%, so we increased it a bit, and so this will increase the number of peptides on the already identified protein. So that's why we call it sequence coverage increase, because it doesn't generate new protein groups or protein sequences, only the known ones that you found in the first search, but you get more peptides on those, resulting in better quantification just because you have more data, and also potentially better resolution of alternative splicing, because with the sequence coverage increase, you might actually add peptides that you didn't have before on new exons or on new splice junctions. Right? So that's a very useful feature of this sequence coverage increase. And so let us look at some examples. So this is just some simple Orbitrap HeLa DIA data with the 3 replicates. And so just if you do the regular search without any sequence coverage increase, you would get these 7,700 so-and-so protein groups. Now, if we do the sequence coverage increase with 1% FDR, we already get more. So why do we get more? Although we use the same FDR as before, we do get more because of the restriction of the due to the restriction of the FASTA file to the ones that were identified before. We remove the possibility of creating false positives in the remaining part because it's not there anymore, right? And by that, you already increase the number of identified protein groups by a couple of percent. But then if you go, for example, to 5% FDR, you get many more protein groups here out of this, and even more so for 10 and 20%. So now to get a feeling for what are these, so why do we have now so many more protein groups than we had before? So do we actually add really new gene models that produce these proteins? And the answer to that is actually not so many, because if you collapse these protein groups to the gene names, to the gene identifiers that come with the proteins, you will see that the number or the difference between no sequence coverage increase and 5% sequence coverage increase is actually not that great. So we can actually get in total almost 10,000 protein groups, but we collapse to gene names, you actually only get 8,000. And so almost none of them are unique to the case where we didn't use the protein groups, and we get a few more from the sequence coverage FDR, but much less than the actual protein groups. So which advocates for that the added protein groups are actually coming from being able to split between the original protein groups and actually resolve isoforms, so resolve splice variants in these existing protein groups, which we see from that we don't increase the number of gene symbols actually much by applying this sequence coverage increase algorithm. Okay, so that's interesting. So then last but not least, the addition of the peptide correlation filter. So what we do here is we look at the intensity profiles of each peptide in a protein over all samples. Then from these, we calculate some sort of median intensity profile. And now we can calculate from each of the peptide profiles the Pearson correlation, or actually the Spearman correlation, with the median profile. And we use a threshold that we only include those peptides that have a minimum specified Spearman correlation. And so by that, we can get rid of false positives in the peptides that belong to a protein group that are either badly identified or actually did belong to another isoform and show different independent quantifications. By that, we should also get cleaner quantification of the protein group under investigation. So yeah, so these were the new additions, and let's look at another benchmark. So the previous benchmark was from timsTOF Pro, and now here we use another benchmark data set, also a species mixture, now a 3-species mixture in different ratios, but this is coming from the Astral instrument, so coming from this recent paper from the Jesper Olsen team. And so just looking at the number of identifications, you can see that that we're doing actually pretty well in terms of the number of identifications in comparison to the reported results in the paper. So the leftmost ones are the identifications from the paper. So the next ones here, these are our identifications. If we don't use these advanced features, then if we filter with the peptide correlation filtering on these results, we get the ones in the middle here, which is almost the same, which means that the peptide correlation is actually not filtering anything if we apply it to the data with 1% FDR. So that, which is kind of nice and expected also, that when we use only solidly identified peptides, actually none of them are filtered out, right? So then if we then apply only the sequence coverage increase, we get a substantial increase from here to here in number of protein identification, as we also saw before in these Venn diagrams. And if we now use the peptide correlation filter on these, this actually does filter out some of the protein groups which are not reported for quantification anymore. So then in this case, where we actually add this little bit of noise and uncertainty to the data by using the sequence coverage increase with 5% FDR, the peptide correlation filtering is actually effective and filters out something, which is also what we expected it should do. So that's all good. So really the identification and quantification is really now on par or better than the other DIA tools that are around that we are constantly benchmarking against. So here you see quantification results of the 3-species mixture. There's also one way how we can optimize the sequence coverage increase, because you notice that you chose a too big value for the sequence coverage increase if you see that the ratio distribution gets wider, right? So that the ratio are not estimated as good anymore in this benchmark data set. So that's how we can actually then come up with the recommended value between 3 and 5% of the sequence coverage increase FDR. Okay, so that being said, then the remaining question is, are these FDR estimates that we're doing also correct? And this we can do by doing a entrapment search where we use humans, DIA samples, and then we offer in silico libraries combined from human and Arabidopsis FASTA files, so that we know everything that is identified in Arabidopsis is a false positive, and we can actually see that our true protein FDR is actually around 0.5% when we set it to 1% nominally. So that means to actually even better than we say in restricting the FDR. So there's no overfitting or any other mischief going on in the algorithm. So it's valid FDRs that we are reporting. Okay, so we have already since long regarding scalability a batch processing feature, which just means that this first computation heavy part where like the feature detection and matching of the library is done in batches, and then whenever one batch is ready, the intermediate results are deleted. And in principle, you could even, although we haven't done ourselves yet, use batch size of 1 which means you just process one raw file while it's coming from the machine, and then collect the results, showing away intermediate results, and collect like the information needed for quantification in one place where then the quantification is done. Right, so you can download. So we have now version 2.6.1. 2.6.2 is coming out still this week with a lot of speed improvements, actually. So that might also be interesting to wait for that. So probably tomorrow, day after tomorrow, it's coming out. 2.6.1. So today is actually wrong. So this is already out for 2 weeks or something like this. And so what are we working on? So next releases will see PTM localization from DIA data that is close to being finalized, and we're still working on this processing speed, which I mean, one can always work on that one, I guess. And so by using some trick of indexing peaks, as it's also used in MS Fragger, for example. And by that, I'm done. So I want to thank you for your attention. And I also want to note that we are going to have a summer school here in Munich, or in Munich. You're maybe not here, all of you, but I am. And this is going to be during the second week of September. It's being in the center of Munich, and registration is open on Eventbrite. So there are still tickets for sale. And it would be great, of course, to welcome one or the other of you to that event as well. And with that, I'm really finished now, and I'm happy to take questions if there are some.
Thank you so much, Dr. Cox. Very, very nice presentation. We have a question in the chat. With the peptide correlation, if you're comparing very different samples, meaning different time point in development, for example, are you not eliminating peptides that are different due to the biology itself and not to the variability?
No, it's exactly the same. So it's looking in the other direction, right? So it's actually looking how these peptides are correlating over all of these samples. So this is like one peptide, many peptides for one protein, which like should actually all behave the same, right? And then it's actually very good if you have a sample where proteins are varying a lot, because then you get a highly significant correlation, or you should get a highly significant correlation. And this is exactly the situation where this filter is working best, if you have like a lot going on between the samples.
Okay. Awesome. Thank you so much. Please get in touch with Dr. Cox, if you have any other questions later. And also please scan the QR code and, spread the information about the summer school. Thank you so much, Dr. Cox. We really appreciate your time.
Thank you.
Thank you all. Thank you all for joining.